super interesting. And again, we'll be
The
going
neutrophil catches up.
over that a lot in other, like when we start to cover the cytoskeleton. Okay, so that's really all I would want you to know about the phagocytosis, specialized cells, eating big things. We're going to go over really focusing on clathrin-coated endocytosis, but it's worth knowing that there are other types of endocytosis that's really a continual ingestion of membrane and fluids from the outside surface. This involves non-coated vesicles, so these would all be vesicles that are non-coated, so that when endocytosis is happening, a vesicle is forming from the membrane. There's no coat, and it's just a continual process that happens. So what do you think would happen to a cell if pinocytosis was blocked, but exocytosis continued? So do you think the cell would become larger? Do you think the cell would become smaller? Do you think there would be no effect? So maybe think about that and think about why you answered your answer. I'll give you guys like half a minute to think about that. So really think about what would happen if you blocked pinocytosis, just this continual pathway that happens in a cell. That exocytosis is still happening. So what do you guys think? Do you think it would be A, B, or C? Yep.
B.
Okay, why do you think the cell would become smaller?
Since it's no longer, something that's releasing more and more particles of a, biologic, be releasing more matter than it will be consuming, so it should.
So exocytosis, remember that you're getting here, and you have these vesicles, right, that are fusing with the membrane. So when that vesicle fuses with the membrane, this vesicle, what happens to the vesicle of what happens to the membrane of the vesicle? What does it become continuous with?
The ER.
The what? The plasma membrane, right? So when this, I'll kind of draw it again. So like if this is the plasma membrane, and that's the vesicle, you're going to get fusion. Maybe I should draw them in different colors.
I said it with a question mark in the end. Shut up.
Okay, so you have this, and now you have your vesicle, and that's going to fuse. And when it fuses, what you're going to get is the plasma membrane, the vesicle membrane, and then the other part of the plasma membrane. Does that kind of make sense? So this will get incorporated into the plasma membrane. Every time there's an exocytosis event, that's the fusion that's occurring when the snares coil upon each other and those membranes fuse. So exocytosis, is that in general making the membrane bigger or smaller? Bigger, right? So if you allowed this to happen, but you blocked pinocytosis, in that case, what would the answer be then? It would be A, right? So then your cell is going to continue to grow because you don't have any mechanism, or less of a mechanism, to bring that membrane back into the cell. So this continual process that we're not talking too much about is very important to maintain homeostasis of cell size. Because if exocytosis was just allowed to occur, that cell is going to actually grow. And it's going to become larger to an extent, right? To the extent that you have membrane availability, internal membrane availability. Does that kind of make sense, everybody? So that's why you would see that. Sorry for some color coding there. Okay, so that's one of the values of these non-regulated pathways of pinocytosis. Again, that's a type of endocytosis. Okay, does anybody have any questions about that? Yeah?
So it's kind of like shrunken looking. Like I understand why the plasma membrane would grow, but if it doesn't have the water coming in, would it get like dehydrated looking?
Yeah, so that's a good point. So it is taking up fluids, but there's also just normal like osmotic changes that I think would suffice for that. So I don't necessarily, I don't know, but I think because of just like natural debility of water, you're kind of naturally passing it out of the membrane to some extent. I think that, I don't know if it would necessarily become like hyper or hypotonic. But that's a good question. You'd have to really test that directly. Yeah, it's a good point. Okay, so this is really getting at now the idea of regulated. So when you think of regulation, this is something that doesn't just happen. You have to have signals in place to allow this to go. And so this is regulated ingestion. And so these in general use what's called Clathrin-coated vesicles. I think you might have learned about Clathrin-coated vesicles in 220, but there might be some people did and some people didn't. I think it's like a newer part of what's included in 1220. So this is an example of the Clathrin-coated vesicles here. Yep, and this is our table. So remember, we had learned about COP2 coats, and we're going to now learn about the Clathrin coats. Clathrin can be involved in things other than endocytosis, actually, but we're going to talk about the Clathrin that's involved in endocytosis. Okay, so one of the things to appreciate is that there are areas of the cell that's called Clathrin-coated pits. And so when you're looking at some of these pictures of endocytosis, we're going to sometimes flip. So now this is the extracellular space, and this is the cytosol. The way we've been looking at it, this has been the extracellular space and cytosol. So try to label it for you so you know what orientation you are at. And if you were to look, if you were to be in a cell and look at the top of, or the bottom of the membrane, you would actually see areas with Clathrin proteins surrounding it, and these are called Clathrin-coated pits. And this might be some of the things you would see. Again, you're inside the cell looking at the kind of cytosolic part of the membrane. This is like what's called a triskelion subunit of Clathrin, but once it's fully assembled for the coat, it forms these really interesting geometrical shapes, and then eventually will invaginate and form a vesicle into the cell. These are all through electron scanning microscopy. And so this picture here is pretty, but it's actually made from biochemical studies that have actually shown the shape of the Clathrin coat. So it's composed of subunits that assemble into a triskelion triple spiral. So you see like one of these. This would be like one subunit, another subunit, and another subunit there. So there's rotational symmetry around these. Yep. The triskelion, so you see in blue, so this is actually 3 protein subunits, and this is the way that they assemble. So this whole three-armed structure is called the triskelion. It's composed of 3 Clathrin proteins. And then the higher order assembly is these units of 3 Clathrin proteins called the triskelion, then assemble to basically form this really cool soccer ball looking type vesicle. Of course, this would be attached to the membrane. So this is just an image showing that it's hollow, but it's not. It would be attached to a membrane. Okay, so they're super cool. I wanted to show this. I don't think it will take that long. So the person that generated some of these movies of Clathrin-coated assembly and pictures, her name is Janet Aissa, and she is a molecular biologist, but she's also an artist. And her goal has been to do a better job at animating biological processes, and those animations are based off of molecular data. So it's not just coming up with something pretty, it's actually using research to generate animations. And my lab at UNC Chapel Hill worked with Janet because my advisor was really into the arts, and he's like, she could do such a better job than we're doing in showing our process. So we recruited her, and she did a really nice job in basically making research come to life for our labs. So I found this TED Talk. It's not that long. I think it's very hypocritical showing that video and then showing you a bunch of things that she just said don't do. But that's all I got right now, so I'm sorry. I'll send my laptop to this weird right now. Okay, so this is what she was saying is not very good. So very sorry about that again. But it's kind of what we got. Plus, this is from your textbook. I do like kind of reinforcing that because it helps if you want to read about the process. Okay, so this is a kind of step-by-step view of clathrin-mediated endocytosis or receptor-mediated endocytosis. And kind of what the process that she showed there is that, again, we're looking here at the outside of the cell, extracellular, and inside of the cell. So that's the orientation. A red dot is something that has to get inside. And it, like you guys, I saw a lot of people drew this receptor binding, so it's binding to some receptor. And then that's going to recruit proteins of the clathrin-coated vesicle. So there's an adapter here. That's going to serve to recruit the clathrin coat. That's going to bend the membrane inward. Remember we talked about bending of membranes isn't the best kind of conformational shape. So these proteins help shape and sculpt that membrane into a bud. If you remember when we were talking about the ER COP2 coats, I showed something that looked similar to this, and we looked at the neck, and we said that it's not really clear how the neck pinches off of the vesicle. We do know the protein that is involved in that pinching in clathrin endocytosis, and that's called dynamine. I have a slide that goes a little bit more into that mechanism. You have now the vesicle, and in the vesicle you have the membrane that used to be on the outside. You have the molecule that needs to get on the inside, down to the receptor. And what's important to know is this idea of vesicles and fusion is basically used again with endocytosis, meaning that this was all great. This helps the membrane curve and get this whole thing inside. But this alone is not really sufficient to do much. It's all really, it's too kind of protected by that clathrin coat. So in that video process, she showed how then there are other signals that come and release the coat so that you, again, have a naked vesicle that can then move on in the endocytosis pathway. And we'll go over what the next kind of spot of that vesicle would be. Anyone have any questions about that? Kind of simplistic, but I think it does a good job showing what's going on. This is the same thing in words, what I just said, so I'm not going to repeat it, but you have that. Dynamin, so that dynamin is yellow protein here. It's what pinches off the vesicle to be like now non-attached to the plasma membrane. Dynamin uses GTP to promote a twisting behavior. That gets the membrane in such close proximity that it actually fuses, and that's how it pinches off of the plasma membrane. So if this is a GTP binding protein, what protein is it probably? What class of protein is it probably? We talked about these a lot. GTPase, yeah, and it is. So this uses GTP hydrolysis to change conformation, essentially. Dynamin is the only protein ever identified that has been shown to kind of wrap around something and like actually twist and twist and twist and twist. And that twisting is energy driven. And eventually the hypothesis is it pulls these membranes so close that they fuse, and that's how you get dissociation of the vesicle from the plasma membrane. And this is cool, this is an EM. This is the clathrin-coated vesicle, so the neck is kind of long. You can actually see the dynamin protein wrapping around the neck, which is kind of cool. I like those, like, very detailed pictures there. Okay, anybody have any questions about what I just said there? All right, so if this is your naked transport vesicle, this is actually going to fuse with another intracellular organelle. Does anyone know what that organelle is? It sounds like it's involved in endocytosis. Endosome, yeah. So if this has to fuse with an endosome, what is missing from this diagram here that has to be on this naked transport vesicle? What's required for fusion?
Snares.
Snares. It has to have a snare. So what type of snare would it be on there? B or T? B, yeah. So there is a B snare. It's just not depicted, but it has to be on there. And then what's the other molecule? Also GTPase. RABs, yep, so there would have to be some other type of RAB protein. I don't know the specific kind, but there would be a RAB on there. So I just want to go over the fact that that is what happens in exocytosis because vesicle fusion is being utilized. Endocytosis, vesicle fusion is still being utilized, and that core process is conserved, whether it's exo or endocytosis. Okay, so that's what's not. Okay, so we just talked about this. Where will I head to next? Endosome. So there's different types of endosomes. They're called early, late endosomes. I'm not going to have you distinguish which one is which. So for this class, just kind of knowing that they go to an endosome is sufficient. But it's worth knowing there are different types of endosomes and different processes that a certain molecule might take. Okay, so this is kind of the pathway. This is where the first fusion event would happen at that early endosome. Okay, so again, I'm going to keep this like relatively simple, but this is showing here that you have endocytosis and a vesicle that's basically going to fuse with the endosome. And it's worth kind of noting that the endosome is also kind of the first stop for these molecules to end up in lots of different places. So one of the places these molecules can end up is actually the trans Golgi. It can be fed back into the Golgi. Or it can be fed through later endosomes and to a lysosome where there will be degradation. Or this can actually be fed up through what's called a recycling endosome, which will recycle things back onto the plasma membrane. So those are all different options that can happen. And that's basically what these 3 points are. So it can direct proteins directly to the lysosome through intermediates. It can recycle proteins directly back up to the plasma membrane. We'll talk about why that's really important when we do the cholesterol example. Or it can communicate with the trans Golgi. Okay, so for this diagram, I'm not going to ask you to name the specific RABs. I don't think anyone memorizes that unless you are actually in a lab that studies RAB proteins. But one of the things I would like to briefly say, and again, you don't have to know this for the exam, but it's worth thinking of, is these compartments are all color-coded. And so if you look at the legend, what do these color codes stand for? What's the difference between these? Does anybody know what these are?
Looks like
So these are all different, slightly different types of phospholipids. So if you look, what's really interesting is the composition of specific types of phospholipids change when you look at, well, the ER, which is not represented here, but the Golgi, early endosomes, lysosomes, and that's actually a way for the cell to know which organelle, which organelle it is, basically. So like this is a signature of Golgi, this is a signature of lysosomes. You don't have to know that. It's something we don't talk a lot about in class, but it's probably worth just kind of thinking about. Okay. So this is a movie of fusion events. It's always kind of fun to see.
In these cells, fluorescent Rab
That's undergoing lots of fusion events, more than a cell would normally undergo. And just this, so this is a really cool one. You can see those two fusing.
observed.
And so they did a trick, which they said in the beginning. So this is more than a cell would normally experience. And this is because they tagged these membranes with GFP, which is what you're looking at. But to basically get all these fusion events to happen, they overexpressed a RAB. This particular RAB is RAB5. So this RAB promotes vesicle fusions. That's one of its roles. But what I wanted you to think of from a molecular standpoint is how might a researcher overexpress the protein? So think about what the researcher may have done to overexpress RAB5. So you can think about that for a minute, and then we'll go over some of the things that might happen to allow for overexpression of a protein. To that protein.
You can increase the promoting, the promoting region, or we can move it to somewhere else where the promoting region is much more active.
To put it under a promoter that's active a lot. And that's exactly what you would do. So have you guys ever heard of a constitutively active promoter? That's what this is. So constitutively active promoter. promoter that's just on a lot, not the endogenous promoter, so this is not the normal Rab5 promoter. And then you simply just put the gene for Rab5 under that new promoter. And then you basically take this whole DNA construct and you put it in the cell. That's a process called transfection. It's okay that you don't understand that or don't know that part. But you would basically create this in a lab in a test tube, and then you have to get it into the cell, and that's how it's going to overexpress Rab5. And so this is a very common way to overexpress proteins for an experimental purpose. Okay, so let's go over now a specific example of receptor-mediated endocytosis. Cholesterol intake, you may have been familiar with this story, so if you are, that's good. If not, it's okay. As you're probably aware of, when we eat foods, part of that important part of eating is getting cholesterol. It's very important for the stability and healthiness of our membranes. Cholesterol, remember, is a vital part of the plasma membrane. It's very important to have good, healthy levels of cholesterol. And so when we eat cholesterol, it's basically packaged in a molecule called LDL, which stands for low density lipoprotein. And that's all basically then put into the blood, the circulatory system. And then those LDL molecules have to be endocytosed into the cell so that your cell can use the cholesterol. Cholesterol can't just freely pass through a membrane. It's actually inserted at the level of the ER. So the question is, though, how does cholesterol move from the blood extracellular to inside of the cell? So, of course, that's a prime example of endocytosis. This is a picture of an LDL molecule, so this is what would be in the blood. What's important to know on your end is that, like, inside of that vesicle is basically where the cholesterol is, the usable cholesterol. The vesicle or the molecule is coated by phospholipids. And there's also a protein that's associated with this molecule called the apoprotein B. So there's an actual protein here that kind of inserts into this molecule here. So it's a single layer of phospholipids, cholesterol inside, and a protein. Yep.
So it's only single layer, or is it bilayer?
Yeah, so this is not a bilayer. This is a single layer of phospholipid. Okay, so what's actually happening for the endocytosis, very similar process that we just went over in the schematic. Again, my apologies, Janet. Probably much better ways to show this. But LDL binds to a receptor. The clathrin-coated pit then begins to assemble that beautiful triskelion. That geometry is going to happen. The membrane will then invaginate. Thymine will come and let the release the vesicle, and then uncoating will occur. You need the uncoating to happen for fusion to happen. If it's like this cannot fuse. So then this fuses with the endosome, so that now this molecule, the LDL molecule, and the receptor are both in the endosome. Okay, so now I have an endosome, and what's interesting, so this is like kind of point number one, the receptor, the LDL receptor, and the LDL molecule dissociate in the endosome. So the endosome has, in general, acidic conditions, and it's going to make this binding unfavorable, so that you have release of the receptor from the LDL molecule. One of the questions that the slide I just skipped over was, if there is a receptor and it's recognizing the LDL molecule, I think it's important to think about what it's actually recognizing here. So do you think the receptor is recognizing the phospholipid or the protein there? Receptors usually are good at recognizing proteins, not all the time, but in this case it does. So this is the importance of this apoprotein B, because this is the protein that the LDL receptor is actually recognizing. Okay, so now these dissociate, and they have two very different fates. So one of the fates is that the LDL molecule will then form a vesicle out of the endosome. So now there'll be another vesicle that forms and is transported. This is all via vesicle transporting, meaning microtubules. Fuses with the lysosome here. And in the lysosome, there are enzymes that break apart the LDL molecule and releases cholesterol into the cytosol, where then it will be taken up and used appropriately in membrane assembly. Really importantly, the receptor does not go to the lysosome. So think about this process and think about the cell always wants to uptake cholesterol. And let's imagine the receptor headed to the lysosome. What's going to be the problem when we look out to the plasma membrane? Do you think it would ultimately affect the number of LDL receptors if this was targeted to the lysosome? Lysosome is going to completely degrade this protein. So if the receptor ends up here, it's gone. It's not going to be active anymore. It's going to be chopped up in little amino acid bits. So the problem is that for the cell to get a new receptor, what would it have to do? Transcribe the gene, and then it would have to translate the gene, then it has to go through the secretory pathway, and that's time, right? That's going to take a good amount of time for this all to happen. So the cell doesn't really want it. That's a work-intensive process. So what the cell has evolved is a way for the receptor to be recycled. So instead of this getting targeted to the lysosome, a bit of the endosome forms a membrane, a membrane vesicle that then fuses with the plasma membrane and returns the receptor to the plasma membrane. It's a really clever way. The cell can save energy by not having to go through that process of transcription, translation, secretory pathway. It basically will undergo exocytosis to go back onto the plasma membrane.
But then it's also taking some of the environment within that endosome back to the cytosol and
Well, so the beauty is, whatever's in the endosome never sees the cytosol, right? So how can this see the cytosol?
Sorry, not cytosol,
Huh?
the extracellular.
Extracellular, yeah, there's going to be a little bit there. But the volume, think about the volume of the extracellular space is so much larger. But yeah, you might get some things that are secreted as a result of that.
But wouldn't that also the environment change the actual receptor itself?
So remember though that this is part of this endomembrane system of which the endosome is a part of. In general, the environment here, even though this is more acidic, is much more similar to out here than in the cytosol. So in general, when we look at lumens of the endomembrane system, there's much more similarity there. So this might temporarily change the conformation of that, but then once it gets exposed, it should refold to the correct conformation. And this is pretty like environment. The lysosome really isn't, because the lysosome is being actively pumped with lots of hydrogen atoms and other hydrolytic enzymes that will basically cleave peptide bonds and other things, yeah. Okay, so does anybody have any questions about that? It's really a clever and cool process. I'm not going to show this video, but you can watch it on your own. I posted all the videos. I didn't, just let me know. I think I did. In my mind I did. To D2L, so you can look this up. Doesn't go over anything that I didn't just discuss, but sometimes it's helpful seeing the information. Okay, and so there's a disease that basically affects this process called familial hypercholesterolemia. These are individuals that tend to have high cholesterol no matter what they eat. And so if a person has this disorder, it's really hard to just manage cholesterol levels based off of diet, although that's an important part of controlling this disease. They also need to have specific cholesterol-lowering medications to lower basically circulating levels of LDL. So when I say high circulating levels of LDL, do you think that means LDL outside the cell or inside the cell? Outside the cell. So this is like blood levels. Circulating means in the blood. And so when you think about potential genes that might be mutated, that's resulting in this disease, what things could be mutated? Receptor, I heard a lot of that, yeah, and that's actually the gene that's most commonly mutated. So in most individuals with hypercholesterolemia, it's the receptor that has a mutation in it. And thus, it has a much harder time getting into the cell. This is a genetic disorder, so this is something that is inherited. You're born with this condition. And so it's something that can be diagnosed, but the molecular mechanism is that the mutation lying in this receptor. Any questions about that? So the gene here is the LDL receptor. And you could see if it can't endocytose, they're going to have higher levels out in the blood. Okay, and so this slide is just saying what I had said, that this LDL receptor is recycled back onto the plasma membrane. And again, this would bud from the endosome. You would have to have the snares here, and then it would fuse with the plasma membrane. So again, the fusion event is going to be really similar. Okay, so I'm not going to go over this again, but hopefully maybe now you have a better understanding of what would actually happen to this red molecule. Good study approach is to do it again. Again, just take a blank sheet of paper and draw it all out, and you can think about this now as maybe the LDL molecule and exactly what would be happening both to the LDL molecule once it's in the endosome and to the LDL receptor, two different paths that those are going to take. Okay, so we talked about how the lysosome is a organelle that is going to be proteolytically degrading proteins and molecules. The lysosome does a lot of work, so it helps with things that are carried from the endosome to the lysosome. But also, when a cell does undergo phagocytosis, often that thing that is phagocytosed will also be headed to the lysosome, and that's how it will safely be destroyed. But another process that's important to talk about is, so I've always been taught, when I was introduced to this word, it was autophagy, but Dr. Sweet uses autophagy, so I'm going to try to use the way he says it, autophagy. People say it either way. I just come from the autophagy camp, and he comes from the autophagy camp, but you guys took 220, he got 2 first, so autophagy. So autophagy is a process that's like so cool that the cell will basically identify organelles or cellular debris that's damaged. It will self-form a membrane around that, which is ridiculously interesting how that happens. So this is all internal. And that autophagosome will then get targeted to the lysosome where those bits will be destroyed. So autophagy literally means self-consuming or self-eating. This is just another picture showing you how these organelles or other kind of bits of the cell